Okay. So we talked about some of the receptor families. We've talked about the mechanism of action, steroid hormone receptors. We've talked about the GPCRs. We've talked about the enzyme-linked receptors. And now we want to talk about how do we turn these signals off, okay? At least in the case of GPCRs, as they have a unique mechanism of desensitization. So when we administer the hormone, or when the hormone binds to the receptor, you get this signaling cascade. But at some point, you want to turn that receptor off, because what can happen is you have essentially an uncoupling from the receptor from the actual signaling cascade. And this can lead to a desensitization of the receptor. And this desensitization means you can no longer cycle. Remember that cycle of receptors. You can no longer keep reactivating the same receptor until the hormone or the drug that you're adding falls below a certain threshold, and then the sensitive can become sensitive again, the system. So prolonged exposure to hormones often results in a gradual decrease in the number of receptors expressed on the cell surface due to internalization and downregulation of the receptors. So after a hormone binds a receptor and you get this desensitized receptor, what happens is the receptor gets internalized, okay, into these endocytic compartments, and then eventually will be recycled. However, after a time, it can get internalized and degraded, and you'll need to make more receptor and translate more receptor to restart the system. So these are two different mechanisms for how we can lose receptor at the surface for stimulation. So in terms of GPCRs, what does this desensitization look like? In this case, you have our receptor still at the surface. A ligand binds. You get that GTP-GDP exchange that I mentioned to you. Then you get recruitment of a family of enzymes called G protein receptor kinases, GRKs. They phosphorylate the tail and the loops of the receptor. This then drives recruitment of arrestin proteins. The arrestin proteins then eventually can lead to recruitment of clathrin adapter proteins, AP2, and eventually you get internalization via clathrin adapter proteins. Then typically your receptor can get ubiquitinated and either lead to lysosomal degradation or that recycling event that I mentioned to you earlier to bring back the receptor, okay? Importantly, all of this occurs within seconds or minutes, and the effects are usually quite pronounced. And then the receptor often will recover when the hormone is removed. This beta-arrestin bound GPCR that I mentioned to you, often the internalization leads to degradation, but in some cases can lead to recycling. It just depends on the receptor, the strength of the cell type, and the strength of the signal. The recycling itself is a slower process compared to the desensitization. So internalization happens relatively quickly, and whereas recycling takes 15 to 20 minutes, and degradation about 30 minutes. the GRKs are a very important family for driving this eventual desensitization. So activation of the receptor recruits these GRKs, and there's about 6 of these kinases. The GRKs use ATP, hydrolyze ATP to ADP to add phosphorylation. You typically add serines, sometimes on threonines, on the tail or the loops of the receptors. Recently, a few years ago now, it was appreciated that arrestins do other things besides just desensitizing the receptor. Arrestin can also serve to recruit other signaling molecules, very similar to receptor tyrosine kinases. Remember the JAK-STAT story? Same thing here. Arrestin can recruit other kinases or phosphatases, which can lead to other signaling mechanisms, okay? And this is what is termed often as signaling bias. You have different pathways. You have G protein and G protein independent signaling. Okay, so let's talk a little bit about quantification of hormone levels. So remember when we talked about Berthold and how he discovered the hormones, right? What did he do? He removed the gonads of the males and he looked to see what happens, right? And then he replaced the gonads with a male and he switched them around and he saw and measured what happens. And then eventually someone isolated the gland and identified what was being released and modified. In that case, it was a sex hormone. And then this is classically how new hormones have been discovered. Okay, so very crude biochemistry has been used over the years. This approach, or relatively simplistic, crude approach of isolating a gland and purifying the protein was replaced with what's called the functional genomic area. And in this case, we use an understanding of gene sequences to identify novel hormones, hormone receptors, or ligands for orphan nuclear receptors. So in this case, for example, we can look at the genome, the human genome, and say, all right, what is similar gene to growth hormone? What is a similar structure to adrenergic receptors? And this way we can start to identify new hormones or new receptors that could be of relevance or of something interesting. Now, in terms of measuring hormone levels these days, typically we isolate plasma and then we quantify plasma concentrations of a hormone and measure its half-life. This allows us to find out how the hormone concentrations are measured. It can help identify genes encoding hormones, and it can help us identify receptor and gene encoding receptors. And eventually, we can investigate gene regulation in health and disease. The problem is, it isn't easy to quantify these hormone concentrations in the blood, that first step. And why is that the case? What's the challenge here? So these are some biologically important substances and their levels. And don't worry, I don't expect you to memorize the regular levels. There you go, my QQ is active. I don't expect you to remember the regular levels, okay? I will give you that on the problem or an exam, and you'll see what's up. So So don't worry about memorizing exactly the levels of the hormones. But I just want you to understand that there are a lot of hormones and a lot of very different detection levels. You have nanomolar and picomolar. So what are some of the assays used for measuring these very low levels of substances? Immunoassays, which some of you may do or may have done in your project, measures the presence or absence of a substance through the use of an antigen or an antibody. Okay, and these different flavors of these are the radioimmunoassay, which is radioactive isotopes incorporated into immunoassay reagents. So you have a radio-labeled hormone, and then you compete that radio-labeled hormone with an unlabeled hormone. Now, this is relatively old-fashioned, expensive, not very safe, and a lot of hormones these days are difficult to radio-label, so it's not used as much. Then you have the enzyme-linked immunoassay, which most of you would have seen or done for some of your projects. And then within there, you have competitive enzyme-linked assay versus non-competitive. So how does the competitive immunoassay work? You have an unlabeled hormone, which displaces a bound labeled hormone, which you then measure. Okay? So imagine we have a fluorescent labeled insulin. We know how much we put in there, and then we want to know how much insulin is in the patient's body. So we take a blood sample, we then put that into our known concentration of fluorescent insulin, and we measure the displacement. And you can run calibration curves so you know the concentration of what you put in, and then back calculate on the concentration of the hormone from the sample. Instead of labeling the hormone, you can also use an antibody if it's available, which will bind the hormone. And this is called a non-competitive immunoassay. And so in this case, you have an antibody which binds your hormone. Then you have a different labeled antibody. It's fluorescent labeled or colorimetrically labeled or an enzymatically linked labeled. And then this binds a hormone at a different site. And then you can measure directly the amount of antibody that's bound to the hormone of interest. Okay? And the signal that you get is then directly proportional to the amount of hormone available. And again, you can run standards. So what are some of the problems with immunoassays? You can get interference, so the hormone may be sticky and bind to the plastic. You may get antibodies that are nonspecific. You may have problems from the plasma interfering with your fluorescence. Peptide hormones, which we'll talk about later, can exist in many forms. They can be cleaved, they can be glycosylated. So which will you be able to measure all of these forms? Some of the hormones are very small molecules, so it's difficult to get antibodies to, or you won't be able to label them. And then, as I mentioned to you, some of these concentrations are very, very low, right? Picomolar levels. So it sometimes becomes difficult in immunoassay to detect those smaller numbers. So more advanced methods are used in some cases, including something called Tandem mass spectrometry, and coupled with advanced forms of HPLC. So in this case, you have liquid chromatography inlet, so that comes in from your column. You put a high voltage on the hormone, it drives it into an inert gas. This just drives individual molecules across these quadrupole, these giant magnets. And then the magnet pulls single ions down and it detects single events so they can print exact mass and ion count for every event that interacts with the magnet. So those are more sensitive. That's a tandem mass spec is a more sensitive method for detecting, but often unnecessary. But at low levels it can be. So the question is, when are these low levels and when do we measure? And what are we trying to diagnose by these measurements? So typically, circulating hormone concentrations are used in the diagnosis of endocrine disease. So a patient comes in, and the first question the doctor wants to know is, what are the circulating hormone concentrations at play? And can we use that for a diagnosis? So obviously an initial question will be, is there too much of the hormone, hypersecretion, or decreased clearance? So is there a hormone, a gland that's making too much because there's a tumor? Or is there a problem where a hormone is not getting processed and cleared appropriately? Or is it a situation where, say, in diabetes, where your pancreas is damaged and you're getting too little of a hormone, so you get hyposecretion or increased clearance? Then the question becomes, what time of day? Do we measure this in the morning? Do we measure this at night? What's the changes? What's known about the changes of the hormone? Remember, yesterday we talked about some hormones are circadian in rhythm and some are impacted by food or sleep. And then, importantly, does it matter that these changes are happening? Do they need to be, is this a hormone that needs to change or not? So this is just an example of 3 different hormones. And you can see you need to know what you're measuring and when you're measuring it, because hormone A here, right? If you measure at noon, you'll see very low levels of hormone. And if you don't know what you're doing, you may misunderstand that actually this hormone should be very high early in the morning. Hormone C is all over the place. And if you don't know that actually hormone C is related to food, you wouldn't get that if you measured it here or here, you might think it's high or low, basically because it's linked to food intake. And then hormone B, right, doesn't change at all, so it doesn't really matter when you measure it. But you need to know that, and you need to know what the static level is and what you're after. And then the question is, well, how do you measure changes, right? So let's take hormone B because it's the easiest one. That's this black line that we talked about. Hormone B is what's called a set point regulator. So it's always at a constant level. And you can measure this hormone level at any time of the day or night because that level should be relatively static. So these are the easiest hormones for the doctors to measure, typically, and are very telling if something is up or down. You know right away something is wrong with your patient because it's obvious. So an excess or an insufficiency is obvious irrespective of the time of day. And these are known as static hormone measurements. So a one-off measurement of a particular hormone will tell the doctor very quickly what's going on. Examples of this type of measurement include thyroid function. So we'll talk about thyroid stimulating hormone and free thyroid, FT4, plasma renin activity, your steroids, aldosterone and androgens, and urinary catecholamines having to do with clearance. All of these can be measured, and often are measured, very quickly when a patient comes into the clinic. What about the hormones that have this diurnal secretion pattern that we talked about? How do we measure those? What do we do for the patient? So these hormones have a set pattern of hormone secretion. So they always have the same pattern during the day. And in this case, hormone A this hormone example, A, will peak in the morning and is lowest at night. So typically what we'll do is we will measure for insufficiency at the peak. So we'll measure here to see and check that it's above the threshold level, okay? In the test for excess, we'll measure the hormone again at midnight when it's at its lowest to check that it's below our threshold level, to make sure that it's changing enough. For hormone C, it's more difficult. So it's very difficult to know when is the right time to measure something like this. So we can either measure every 10 minutes for 6 hours, but that's not very practical, right? Or we can use something called dynamic testing. So what is dynamic testing? Dynamic testing involves using an intervention which should change the level of hormone. Remember I mentioned to you, I mentioned to you that this is a hormone related to food, right? So what we can do is artificially induce those changes by intervening, for example, giving the patient a candy or some juice. So what we would do in this dynamic testing, if you suspect, for example, the hormone is excess, the Nydemic test is designed to suppress hormone synthesis and secretion. Failure to suppress indicates a pathological hormone overproduction of that hormone. This test is known as the suppression test. So, for example, you might give dexamethasone suppression test to see if there's an excess of cortisol being produced. If you suspect an insufficiency or deficiency in terms of production, the dynamic test is designed to stimulate hormone synthesis and secretion. So failure to increase after you stimulate the patient suggests that there's some hormonal deficiency and not enough being produced. This is known as a stimulation test. So in this case, you want to give the patient something that will drive up, for example, cortisol. A common test is this synthetic ACTH, which is testing for cortisol deficiency. You give ACTH, that should drive the adrenal glands to make cortisol. So here's some examples. So here's a circadian pattern in human growth hormone. Okay, the normal pattern, right, in black. Human growth hormone should see these peaks in the evening time. But someone with acromegaly who has a growth defect has much more static levels of growth hormone that are elevated at almost all hours of the day. So overproduction of ACTH is seen in a condition called acromegaly, that you'll hear about. In this case, a single point hormone measurement is not that informative, right? Because imagine you measure when you're just measuring at some of these peaks. So you'll have to check it multiple times to check growth hormone. So to do that, what we can do is one of those interventions that we discussed. So we can give the patient glucose and then measure growth hormone levels. So increasing blood glucose should drive down growth hormone secretion, whereas decreasing blood glucose should stimulate growth hormone secretion. So we can do either one of these, and sometimes we'll do both, to measure and test the patient. So this is called a glucose tolerance test for excess growth hormone. Give the patient a little bit of juice or candy, and then you see what happens. Or you can, this is after overnight fasting. And then if GH is detectable at 60 minutes, then suggests acromegaly, if there's excess secretion of the growth hormone. So this is normal GH regulation, okay, after you give a little bit of glucose. But if it stays elevated, the patient may suffer from acromegaly, okay? So this is just an example of intervention and the type of testing that we can do. Okay, so today we've talked about steroid hormone receptors. Remember, they are going to cross the plasma membrane easily, going to bind and interact with receptors located in the nucleus. They're going to gradually drive gene transcription and translation. So their initial response is slow, but gradually it's quite strong. GPCRs, their initial response is faster, but eventually their gene transcription translation is slower. Enzymatic, enzyme-associated receptors, the phosphorylation is that dimerization event. And then the receptor desensitization and downregulation that we talked about by arrestin. And finally, we've talked about the importance of thinking about the hormone levels, what time of day that you measure those levels, and what type of changes you expect. So, as I mentioned to you, one of my favorite questions is comparing and contrasting GPCRs and receptor tyrosine kinases and giving me examples along with their downstream tissues and implications. And then be able to, in particular, talk about how you measure these changes in hormones and the challenges therein. Okay. Now let's see if I can get you to a Mentimeter again. Let's see if this Mentimeter is going to work. Can you see that?